Thank you for the introduction. We are having in Kubrick in Cardiff in the UK, we have the second connectome scanner after the one here. So we're doing, trying to explore a lot of what new kind of signals these strong gradients can give, and also explore some applications. So I will talk to you about this work that got published in NeuroImage this month on combining free waveform diffusion encoding, so non-conventional diffusion MRI with strong gradients. So in biomedical or biophysical modeling of the diffusion MRI signal, we often consider that there are different compartments within each voxel, and then the total signal can be written as a signal. Can do it with the mouse here. It can be written as the sum of the signals in these individual compartments. And each voxel in the brain is built up in a different way. So for example, in the CSF, we have mostly big spherical compartments, and a partial volume of CSF and gray matter. There might be bigger spherical compartments and smaller ones. And in partial volume of white matter and CSF, there might be some highly restricted anisotropic compartments, or you can have, for example, crossing compartments in certain areas of the white matter. And today I will be talking about these kinds of compartments that are isotropic and very small and dense, and if we can detect those with diffusion MRI. And we sometimes refer to this compartment as the dot compartment because it's simply so small compared to the diffusion length that it almost looks like a dot where the diffusion is just completely restricted. So to go a bit back into the literature with conventional diffusion encoding, where you give pulse gradients, we also call this linear tensor encoding sometimes, but it's the conventional statistical tenor experiment. And a couple of researchers have tried to look at this dot compartment or the detection of that. So here's one work in vivo with 60 millitesla per meter gradients that investigated the inclusion of such a dot compartment and how it could explain the data. So the landmark of a dot compartment is if you go to stronger gradients or higher B values, your signal starts to plateau. And this was clearly observed in some of their data sets. And they found that including a dot compartment simply explained the data that they acquired better than not including this compartment. So they included a dot compartment in their biophysical modeling. But then if you look at most of the diffusion MRI models, for example, Charmed or the standard model of diffusion or other models, most of them do not include such a compartment. And this work that looked with a little bit stronger gradients and much higher B values at the signal decay also found that this dot compartment was actually almost negligible. So they recommend to not include a dot compartment, at least for white matter biophysical modeling. So there's a little bit of a controversy whether we should include this compartment or not in our modeling. So isotropic diffusion encoding is a tool that can make your signal more specific to isotropic highly restricted compartments. So let's say we have a scenario in which there are no dot compartments, but we just have sticks that are a little bit dispersed. Then if you measure in the direction from top to bottom here with conventional diffusion encoding, so you only look at diffusion, for example, in this top-down direction, it will be very hard to disentangle from a scenario in which there are some dot compartments, because you have a little bit of restriction from each of these sticks that are not perfectly aligned with the gradient direction. So it can seem like there is a very slowly decaying signal at high B values. But if you take the same scenario with isotropic encoding, where you are sensitive to diffusion in any direction, then you might disentangle these two scenarios at high B values. I will illustrate that here again. So let's take again these two scenarios, the linear tensor encoding and spherical tensor encoding. And I can come up with a scenario where you have only these dispersed sticks here in blue, and you have dispersed sticks and dots here in green. And they, with linear tensor encoding, if you go to high B values, they almost give the same signal. But then if you use isotropic encoding, at high B values, you can start to disentangle these two scenarios from each other. And if you have a perfect dot compartment, at high B values, any other compartments that are there, they will become suppressed because there is diffusion and you're sensitive to diffusion in any direction. So the signal from those compartments will decay away very fast. And then if you assume that the diffusivity in this dot compartment is, the apparent diffusivity is 0, so the diffusion time is much higher than the diffusion length, and then you will see that this plateau value here is actually equal to this dot signal fraction. So this might, the signal at this high B value might become a direct marker of the fraction, the signal fraction of this compartment. And this is of course limited by the noise floor. So if we take the magnitude image, you have a Rishon or non-central Chi noise, which means that even if your signal is truly 0, it will be moved upwards artificially because of this noise floor. So that means if you have a lot of, if you have a high standard deviation, this upper limit of the dot fraction that you can actually measure becomes lower. Sorry, it becomes higher. So there was a previous work that looked at this dot compartment with spherical tensor encoding or with isotropic encoding. Those mean the same thing. So basically what they did is they applied a lot of pulse gradients. It's a bit more complicated than the simple scheme here, but what you would do is apply a lot of pulse gradients along all the 3 axes before you make your measurements, so that you have sensitivity to diffusion in all directions. And they found that they went up to B values of 5,400, and they found that there was no plateau. So they said to not include a dot compartment in white matter modeling based on this acquisition. But with a clinical type of gradients, the B values that you can achieve within a given TE are lower. So we tried to, inspired by this work, we wanted to repeat this on with strong gradients and use isotropic encoding, making a few improvements to the way the encoding is done. So one thing we did was, instead of attaching to this pulse gradients, which takes quite a lot of time, we switched to free waveforms where you have a more smooth trajectory in gradient space or Q space. We also adopted asymmetrical waveforms, which allows you to shorten this second part after the 180-degree pulse, so that if you have your EPI readout here, you can squeeze that a little bit so that the echo time becomes shorter, which means higher SNR. And then with the strong gradients, we could go up much higher in gradients, and so higher in B value. And we also wanted to look beyond the white matter, so look at other tissue types in the brain, such as gray matter and the cerebellum. So these are the acquisition parameters. We went with quite big voxels, which is similar to the previous study I showed. We went up to much higher B values, and our echo time was relatively short. If you compare that to, there's a standard scan, which is a 1-scan trace image that you can do, and that makes use of these pulse gradients, and that would give you an echo time of 270 milliseconds. So we managed to significantly reduce the echo time. We acquired spherical tensor encoding or isotropic encoding, and linear tensor encoding, which is the standard statistical tenor encoding. And here are the waveforms. So you can, these waveforms, they have like also a broader frequency spectra, which we denote here. But if there's no time dependency in the diffusion process, any spherical tensor encoding waveform at a certain B value should give you the same signal. But if there's time dependency, then of course that starts to play a role. So the processing, we did the Riccian bias correction to get rid of the noise floor and susceptibility distortion correction. We used the MP-RAGE to segment out these different tissue types. And then based on these segmentations, we manually drew ROIs to make sure we were not near any susceptibility artifacts or other artifacts that might artificially give you a high signal at high B values. So here are some of the results. The different tissue types are highlighted with the outlines. And what's interesting to see is if you go to higher B values, you lose almost all the signal almost everywhere, except for the cerebellum gray matter. It really shows up as a white blob almost in the images. If we look at that a little bit more quantitatively, so plotting the B value and the signal for all these different tissue types, these are represented with different colors, as you can see in the image here. Then we can see that in the medial white matter here and in the deep gray matter, the signal really just plateaus at the noise floor. So we cannot distinguish that signal anymore from zero. But in the occipital white matter and the cerebellar white matter, the signal is significantly above the noise floor. So there's still remaining signal there. And then in cerebellum gray matter, there is just a lot more signal still there. So after bias correction, we can study the signal a little bit more quantitatively. So this is what we used to fit our models. So if we go with the assumption of a complete dot compartment, we would expect this plateau here. But as we can see, for none of the compartments except the ones that hit the noise floor, the signal is really plateauing. But still, we can use this signal and the highest B value as some kind of upper estimate of this dot signal fraction. And if we do that, we see that there is, if you assume a complete restricted isotropic fraction, it's about 2% in the cerebellar white matter, and about 0.5, 0.3, so very low at this echo time in the white matter. And the previous upper limit was in the order of a few percent, so we improved that by a factor of 10. So no dot compartment in the white matter, except maybe, yeah, very low percentages. And this is just an image of this fraction in 5 different subjects in the cerebellum. So it seems to be quite reproducible across subjects. So since the signal is not plateauing, there might be a better way to describe it. And that's if you go to very high B values, you can characterize this by not a complete dot compartment, but a compartment with finite diffusivity. So that's what we did here. We just took the high B values where we know we suppressed everything else, like the axons or extra-axonal space should be largely suppressed in that regime. And we can study specifically the signal at these high B values. It's kind of nice to look at it in that way, I personally find, because if you model the whole signal across all B values, you have to model all the compartments. And for example, if you make a wrong assumption for one compartment, it might bias all the other parameters. So it's kind of an alternative way to look at these compartments. So when we allow for a finite diffusivity, we can see that there is a compartment in the cerebellar gray matter that makes up almost 10% or more of the signal there. And in the white matter, this is a bit lower, but there is still some evidence that there is an isotropic compartment with low diffusivity there. So this is again across 5 different healthy controls. So it's really reproducible across healthy controls. This is a scatter plot. So each, for 5 different healthy controls, so each point is a voxel there. So you can see that these estimates are quite, the spread is relatively low in the cerebellar gray matter, which is the blue one. But if you look at the occipital white matter and the cerebellar white matter, then there is a much larger spread in these estimates. This is to visually appreciate the difference in signal that we get from conventional diffusion encoding and spherical or isotropic encoding. So at very high B values. So here with linear tensor encoding, we still see a lot of signal. You also get signal from all the from intra-axonal water, mostly. If the diffusion time is longer than the diffusion length, this becomes like a stick, and you will never lose this signal if you go to very high B values. And then for the spherical tensor encoding, you suppress all of that, and you're really only looking at isotropic restricted compartments. And this is just re-windowing that signal to appreciate a little bit more that there is still significant signal. Here, the comparison between linear and spherical tensor encoding a bit more quantitative. So in all these different ROIs, we found a much higher signal with the linear tensor encoding. So that means they really provide some complementary information. We also looked a little bit in the cerebellar gray matter at the T2 of this compartment. So our echo time was relatively short, and we could also measure at longer echo times where we left the timings of the waveforms intact. So we just delay the readout. And you can see that there is the estimated fraction is different for different echo times. So if you then estimate the T2 of this compartment, it's about 62 or 63 milliseconds. So that's longer than the T2 of, for example, myelin water, but shorter than some of the reported values for intracellular and extracellular water. We use strong gradient and optimized waveform to search for the dot compartment. We found that in agreement with other studies that the signal fraction in white matter is really low. And we found a significant but still decaying residual signal in the cerebellum gray matter with isotropic encoding at high B values. And there are some other studies that looked at at different waveforms, spherical tensor encoding and linear tensor encoding, where they also found a significant signal ex vivo in the cerebellum. But this was done in a different way, where you look at the different frequency spectra. So you play around with how these waveforms are represented in frequency space. And in that way, you can probe the restriction of that compartment. So we know that there is a lot of restriction in the cerebellum, but where does this signal come from? Because it's a really interesting structure. The cell density is so high, and you have a lot of different cells. So you have Purkinje cells and granule cells. They are the most famous. Then these Purkinje cells, they have a lot of spines where water can be trapped. A lot of other cells. So here you can just see a Nissl stain, which shows the cell density. It's really extremely high. So it might reflect water from inside cell bodies. When we use, again, the same signal to get a kind of a size estimate of this compartment, we found that the size estimate is about 4.5 micrometer. But note that these waveforms were never really optimized to avoid, for example, exchange regimes or anything. It's just something that we tried. And the fraction was about the same, but the size was, yeah, about 4.5 micrometers. And if you think about the cell sizes, so the Purkinje cells, they are very large, much larger than this. And the granule cells, they are about 7 to 10 micrometers. So that's also larger than what we find. So it could be that you have a very high, that's actually shown in this next slide. So if you forward simulate signals from a spherical compartment with different diffusivities and different radii, and we found about an apparent diffusivity of about 0.1, so that's this red line here, then you can explain the same signal with a spherical compartment that has around this radius or a very low intrinsic diffusivity inside the cell, so there might be a high viscosity. So it might be that the signal comes from inside the cells with a much lower diffusivity, or it might be for, outside cells that are very densely packed. So also the extracellular space actually becomes very restricted, or maybe a signal from within these dendritic spines. Another discussion point is that we had very low resolution to maintain the SNR, 4x4x4. And we tried some super resolution approach where you acquire relatively thick slices in different orientations, and then you can reconstruct the signal in much higher resolution. But it would be very interesting. I know your group, your institute does a lot of work on high resolution imaging, so it would be very interesting to look at this cerebellum, which has so much structure and foldings, and maybe combine it with this technique where we can be more specific to certain layers. Next steps, we would like to include multiband and optical motion tracking. So the registration of these images was very challenging because mostly there's no signal to base any registration off of. So we acquired interleave B0 images to find some spatial correspondence between the images. And we didn't observe a signal plateau. And so the finite diffusivity is one reason why that is the case. Another reason might be that you have a perfectly restricted compartment with zero effective diffusivity, but this compartment is in exchange with its surroundings. So here's just a little simulation showing a perfect dot compartment, but with exchange. And if you have exchange, then you lose this signal plateau. But we cannot tell at this point whether it's diffusivity in a completely restricted poor, or whether it's exchange effects. And the question is if this can be a new marker in pathology, because with all these cells in a healthy subject, if you compare that to, for example, patients with movement disorders such as spinocerebellar ataxia, these cells are targeted in a very specific way. And we scanned patients with different movement disorders in collaboration with clinicians, where they had a very specific question whether the cells are actually affected in the cerebellum, or is it just atrophy and the cell packing and the cell properties remain the same. So what we could see in this spinocerebellar ataxia type, so the 6 and the 2, is that the black dots are all the healthy controls, so they are very reproducible. These are different lobes, also these 3. And then when you compare that to the signal from these patients, you can see that they significantly deviate from the distribution that you might expect in a healthy population. But this is still work in progress. So this was the presentation about the paper. I do have a couple of other slides about projects that we're doing on the connectome scanner in Cardiff, if there's still time, but maybe there are some questions at this stage.
You know, when you're calculating the p-values for the spherical tensor encoding or linear tensor encoding, those waveforms are so funky that it must get really hard to, really get an accurate p-value, even if you're doing the full. Because when you do the integration with the improved, eddy currents and stuff like that, you have to start wondering if you're really comparing things, apples to apples.
Yeah. So yeah, the eddy currents, we didn't adjust the p-value for that. We did adjust the p-value for gradient nonlinearities. So they are not obviously as precisely 15,000. So we did take that into account in the estimation. We also optimized the waveform so that Maxwell terms would play less of a role. But yeah, it is very challenging to, especially with the high gradients, to see.
But like you said, I guess most of the stuff that's moving faster is gone. So I guess you're getting rid of the time element of that calculation you're doing.
Yeah, you mean the frequency properties of this?
Yeah.
Yeah. So so if there is, so for the estimation of this time-dependent compartment, we took that into account. So the gradient nonlinearities and how that affected the time course of the waveforms, but not the eddy currents, because that's we could do that probably. Yeah, that's going to be really hard actually to. But we try to do it with the information we had, but still the signal is there. So.
Is there enough SNR to see anything outside the cerebellum? Any kind of accuracy?
Only in the occipital white matter. So if you just take the average of the highest b-value images, that's where you still see some signal. Maybe that's because it's just close to the coil. So it might be there, but it might be everywhere, but that we just don't have the SNR to detect it. What we know for certain is that the signal fraction is very low at this echo time.
You laid out a number of kind of hypotheses in terms of the underlying anatomical structures of the signal. In your own mind, is there a class of experiments that you've thought through that will allow you to kind of pin down which of those is most likely, or might there be patient populations where some cell populations are going to undergo and stereotypic ways that you can use to help answer those questions?
Yeah, so with the patient populations, that was one route that we wanted to take to see if patient populations have known cell loss. Can we relate that back to this measure? Another thing that we're looking at is diffusion weighted MRS. So where you look at metabolites instead of water diffusion, and from those you know if they are inside or outside of cells. So you can at least disentangle those scenarios. No, we have done some diffusion weighted MRS, but not combined with free waveforms yet. So at high gradients, we will present that at the ISMRM in Sydney.
Are those even bigger voxels?
Yeah.
10 by 10 by 10?
Yeah.
You knew the answer to that question.
Well, no, because once you get to those really high p values, because NAA has a much lower diffusion than water.
Yeah,
So you might actually start to get comparable in terms of signal noise.
Yeah. But things like fat suppression were quite challenging in cerebellum, so there are definitely some challenges there.
And so at this point, I mean, it seems like you're really just at the beginning. Do you have a best guess, or what's your hypothesis in terms of where the signals actually?
I think it's either from inside these spines or perhaps extracellular space that just becomes very restricted. Sorry?
It'd be awful interesting if they were saying on the spines.
Yeah.
I'm curious what kind of coil you used and how you combine the channels. Does it make a big difference on the receiving bias?
Yeah, so we used the Siemens 32 channel head coil and with the adaptive combine. Yeah, so just the standard. So
you have no idea what's happening.
Yeah, so actually in the paper there is, we did some experiments looking at the noise distribution, and it looks pretty Rician. So we looked at different locations outside and inside the brain, trying to estimate what the noise properties are.
So with a combination system like that, it's doing very different things outside and inside, and that's a function of those signals. So
Yeah.
That's like a really hard method to figure out what's happening. It's much easier in coil combination methods that allow you to keep track of some degree of what's happening. Recent distribution is a special case of a single channel. You have a multi-channel coil. It's not a recent distribution.
Yeah, definitely. That's a good point. I think if we would go back to the raw data and try to reconstruct this in a more careful way, you could do all these corrections better. We just tried to do the best we could, and I believe that there are some papers showing that in certain circumstances with this adaptive combine, it does converge to approximately a Rician distribution. But I think, yeah, the suggestion of keeping careful track of the noise is definitely something we should do, especially when you look at such low signal. Yeah.
It's pretty important to you to know where that noise floor is, so you can determine that it's plateau.
Yeah.
You really need to know. So
I think for the cerebellar gray matter, we can say with pretty certain that it's above the noise floor. Yeah, the other ones, yeah, I mean, the noise floor, that is definitely like the upper limit that we can measure, and probably a bit above that. So that would be an upper limit estimate.
So for the asymmetrical gradient waveforms that you use, did you run an optimization or something to really try to optimize something, because they look very particular?
Yeah, so so this was in collaboration with the Lund group, and they have a tool where you can optimize these waveforms. So you maximize the B value given the timings. Yeah, and then you can take into account the maximum slew rate and things like that.
You still constrain them to have zero sum, right? Zero integral.
Yeah. All right. Yeah. All right. So this is a little bit of just an overview of some of the projects that we're running on the Connectum scanner in Cardiff. And if you're interested in some of these, please come and talk to me, and I'd be interested to chat more. So we did a bit of work on combining diffusion with relaxation. So mostly focusing on T2 relaxation and T2 star relaxation or susceptibility. And because if you look at the microstructure, here is a slice in the corpus callosum. These different compartments don't only have different diffusion properties, but also different T2 properties. And this is a CSF voxel. So if you look at this two-dimensional space, they are actually in different parts of that space. So adding these dimensions might give you extra possibility to disentangle these compartments better. So if we just zoom, we leave out the CSF because it's quite far away in this space, and we zoom at the other compartments, and we plot echo time versus B value, we can see that they have, this is a simulation, but they have quite a different signal profile, where in this space you are and how much signal you still have. So the mile in water at the echo times we can measure, it's almost completely gone, like at very low echo times, and they have very low signal fractions. So the signal is very low from that compartment. If the intraxonal space is really a stick, you will not lose the signal in B value if you measure perpendicular to it. And this is the extraxonal space represented by finite diffusivity. So you will lose it if you go to higher B values. So usually the conventional experiment is on a clinical kind of scanner. If you want to achieve high B values, you have to leave the gradients on for quite a bit of time. And you are approximately able to sample this space. So you can, you start, the lowest echo time you can achieve is already pretty high, so you don't pick up any myelin signal. You pick up some intra and extraxonal signal. Then with the strong gradients, you can significantly extend that space. So start measuring at much lower echo times and actually do this echo time dependent experiment. And then here we have this EPI readout, and we can see that because you have this readout part here, you lose again a little bit in echo time. So my colleagues Umesh Rudrapatna and Lars Muller, they have been working on implementing a spiral readout on the connectome, where you can start reading out just immediately after the gradients, and then you can extend this measurement space even further. We use the field camera to correct these trajectories. So if you then look at what echo time is achievable for what B value, these solid lines is without spiral readout for 40 millitesla per meter, 80, and 300. And then if you combine it with the spiral readout, you get the dashed lines, and we can go to echo times as short as below 20 milliseconds and about 30 milliseconds for a 6,000 B value. So here we acquired that data, which was presented at ISMRM last year. So very short echo times for B value of 1,000, and for B value of 6,000, we had an echo time of about 30. So the question is, can we be sensitive to diffusion of myelin water? This is a simulation with the acquisition settings that we use. And this is the myelin compartment with low T2 and low perpendicular diffusivity. So the perpendicular diffusivity, if you look at the literature, it's not expected to be completely zero. because it can still move around in these shells. And if we look at a location in the human brain with this acquisition, we can almost distinguish the three compartments, but the lowest T2 compartment still has quite a high T2 than when comparing to literature, where sometimes the myelin water T2 is estimated as short as like 10 milliseconds or something. So maybe we're not quite there yet that we can measure diffusion of myelin water, but there is some indication that we have some sensitivity to this. Then another project which we will present this year at ISMRM is together with Elena Kleban. We have a tiltable coil in Cardiff that we could measure within the connectome. It's a 20 channel head coil. So this coil allows you to tilt by 7 degrees and 9 degrees and 18 degrees, and was originally designed for patient comfort, so that they would be more comfortable when tilting. But we try to use it to look at T2 and T2 star anisotropy. So it would make the experiments where you effectively have to rotate your head with respect to the main magnetic field, hopefully more reliable and more comfortable. Then the combination with diffusion susceptibility. So we have the diffusion process, and if we apply, if we have the myelin sheet introduces this really interesting susceptibility properties. If you, depending on how your fiber is oriented with respect to the main magnetic field. So doing kind of like a joint modeling of those two is something we have been working on. So this was also presented at ISMRM last year. So what we did is we introduced this extra parameter TD, the delay time. So this is based off of the asymmetric spin echo sequence, where you delay the readout and you're getting sensitive to these susceptibility effects. And we try to estimate the frequency difference that arises between the signal in the intra and the extra-axonal space, according to this hollow cylinder model. Then Sila Gensch and Erica Raven, they really spearheaded pediatric imaging on the connectome scanner. And so we scanned a lot of kids between 8 and 18-year-olds with an advanced diffusion imaging protocol. And we spent quite a bit of time trying to optimize the pre-processing for this challenging group to scan, because obviously they move more than adults. And we also were quite interested in seeing what can the information from the connectome scanner contribute to investigating the process of aging. So here we looked at different B values and an analysis method called apparent fiber density. So this is based on spherical deconvolution techniques, where you try to look only at the interaxonal space and estimate the fiber density per fiber population. And this also was proposed to work in crossing fiber areas. So the preprint of Scylla is out on bioRxiv if you're interested, but the interesting thing we found was that at high B values, so if you just look at, again, the high B value regime and not at the other B values, it seemed that the age relationship that we could find using this method was much more apparent than with other lower B values.
What happens in the cerebellum?
Oh, we didn't look at the cerebellum here, but we have the data, though, so it would be interesting. Yeah, so that was it. Thank you very much for your attention and for the invitation.